Example 3 of thiamine pyrophosphate is in the context of pyruvate dehydrogenase complex. So when we look at this flow, pyruvate, right? We know if it was going to ferment in our body, what would pyruvate be converted to? We already talked about that. Be converted to lactate plus NAD plus, and that NAD plus would go back to run glycolysis. But that's not what we're doing here, okay? If you're not running lactate or not much at all, then you got a lot of pyruvate being converted to acetyl coenzyme A, which will go into the citric acid cycle. So that right there is pyruvate. Hopefully we've got pyruvate down very, very readily. And then coenzyme A, right? It's going to pick up the acetyl group to form acetyl coenzyme A plus acetyl group, acetyl coenzyme A, and then NAD plus to NADH. Okay, and so there is the acetyl coenzyme A. So let's talk about that. So first of all, the pyruvate dehydrogenase complex, it's got TPP. Of course, that's why we're talking about it in this slide set. Look what else it also has. What's this right here? What's this molecule? What do you think? A, so A is going to be in the name. And then it's got this, right? So what is that? That's a flavin, right? That's FAD. And so when we look at this, so FAD, what's this? NAD plus. What's this? Acetyl coenzyme A. And then this is the lipolysyl arm, which we haven't talked about. But you should be able to identify these now with no problem, as well as where the reaction goes. Here, it's the acetyl coenzyme A area. It's this ring right here with positive charge, and it's this region right here of the FAD. Remember, it can take 1 electron or 2 electrons and 1 proton or 2 protons. So if you still don't have that down yet, make sure you get that squared away of where that's the reactivity. So TPP. So now when we look at this again, let's go back to our pyruvate decarboxylase as an example. And let's see what we got here. So first of all, the reason why this is a particularly good example is if you look at pyruvate decarboxylation to form this right here, right, this carbon ion with the 2 carbon skeleton on it, this is shorthand for that. So these steps occur in the pyruvate dehydrogenase complex, and that's what happens there. So by knowing pyruvate decarboxylase mechanism, which you will know pretty well, we also know the first set of steps for pyruvate dehydrogenase complex. Okay, so now what do we need for pyruvate dehydrogenase complex? It gets pretty complicated. The goal is, right, we need to take this unit off and add it to the sulfur group. So this then is going to attack this lipoamide. So this lipoamide is this thing right here. So you see how this arm fully extended. So this right here is the peptide backbone. And then we've got 1, 2, 3, 4, 5 CH2NH carbonyl group, and then 1, 2, 3, 4, 5. So this is a whole thing that we haven't even talked about, but this is a long prosthetic arm with this functional group here. So we've got this, right, disulfide bond here. It's not cysteine, but it's a disulfide bond. So remember here, when we had this resonance-stable carbanion and pyruvate decarboxylase, it pulled a proton off something to get protonated. That was the job of that. Here, the job is to attack this sulfur, which then breaks that sulfur-sulfur bond. And of course, we can draw that, right? Because we see that that gets protonated. Now, this electron rearrangement, let me just see what I got set up here. I got this on multiple slides. I don't want to get too carried away. So what we're going to see, though, is that this is very similar to what we've seen before. Okay? So in all of these, right? So if you look at this right there, so just forget about this right now. When you go to this, what is this going to form? What will this form right here? Pull off that with the base. Electrons go there. That bond will break. It's going to form an aldehyde group, right? So, you know, we're seeing commonalities time and time again. The goal is to get it squared away. And so then we've got this aldehyde group, and look at what's this? What is this? Right? C double bond O, S, CH2. What are we going to call that functional group there? It's very reactive. Thioester. All the stuff we talked about thioesters, boom, we need to know that now. Very reactive, right? Good leaving group for all the reasons we talked about. Sulfur atom's big, electron density it can exist in, right? It is not as stable as an oxygen ester because there's not as much overlap of the orbitals. And so that is further expanded on here. I may have already talked about everything, so I don't want to just keep going over the same thing. And we did. We covered everything. But what you can do is you can go through, if you're drawing down on the other side, you can draw through this. But let's just go through it one more time. This step is those steps of the pyruvate decarboxylase mechanism. Instead of attacking and pulling off a proton to make acetaldehyde, because if you did that, that's what would happen, it then attacks this disulfide bond of the lipoamide arm. What this allows us to do is then pull this proton off, form a carbonyl group, and break that bond. So that's one thing we should mention. Those first steps are make it, break it, make it, right? And so when we look at this, it's the same idea here, right? We did that, and now what we're doing is instead of a carbon-carbon bond we're making a carbon-sulfur bond, and then we're breaking a carbon-carbon bond. So it's still, every bond you make, you got to break, right, when it comes to the carbon-carbon or carbon-sulfur. Okay, so now this is where we can, you can spend a little bit of time kind of comparing them. Again, very similar. For a third for all, there's electron rearrangement, which results in carbon-carbon bond breakage, right? So where are we for the pyruvate decarboxylase for this, right? This right here. So we know that it starts out here, right? So attacks the carbonyl group, right? Electron rearrangement results in carbon-carbon bond formation. Okay, so what is the leaving group? It's the same thing that we saw before. The leaving group is the carbon ion and eliminated from substrate carbon dioxide. So this is for PDHC and what? What else can we write here? Pyruvate decarboxylase. And so this is where it gets different, right? So now what we have is, after we go through there, now we've got this, this carbanion right here, right, with resonance. So this right here is what's happening for right there, right? This is this, same exact thing, right? So what's going to happen? This is going to attack that sulfur atom that's engaged in disulfide bond in that lipoamide arm. Boom, right there. So what's the leaving group? For that, if you're going to break that bond, what's the leaving group? It's that sulfur. So that sulfur, that thiolate, well, we know that it's going to get protonated, but still, the leaving group is the sulfur. That's where the electrons go to, and we've got that carbon-sulfur bond formed. Okay? And then we have electron rearrangement. Again, we know that there is a base pulling that off, so we could put base catalysis instead of just electron rearrangement. We definitely need to know that, that a base is acting on that, right? So base pulls that off. And then what we have is carbon-carbon bond breakage, and the leaving group is the carbanion. Right? It loves those electrons. It's an electron sink. That's its job, to pull those electrons, to be very reactive. And then now what we've got is this thioester, right, which has this acetyl group on it, which is ready to be attacked by the sulfur of the coenzyme A, to then essentially it's exchanging the sulfur groups, right? So this is a good leaving group, but really when you look at the energetics of this, you're trading, this bond right here is really no different than this bond right there, because it's not, it's the same thing. Right? So it conserves, by making this, it conserves the energy to then be essentially at this point then, it releases it. And then there are other steps because we need to work on that, but we'll get to that in the pyruvate dehydrogenase complex slides, not this. We're just focusing on the TPP in this one, right? So that's the function of this. So there we've done it. We've just covered these 3 examples. We see that the pyruvate dehydrogenase complex, the first set of steps are the same exact steps as the pyruvate decarboxylase. It's just that instead of abstracting a proton for that carbon ion, right? This is instead attacking that sulfur of that disulfide bond of the lipoamide.